Well, hello. I hope everybody's having a good day. As you know, I'm in Cincinnati at the Society of Sensory Sensory Science Professional meeting. Sorry to not be here. Hope you did all right on the exam. We can talk about that on Tuesday. They should be graded by the time I come back. So, I want to pick up where we were. We were talking about collagen, as you know, one of my favorite topics. And we had gone through the structure of collagen. We talked about glycine, the amino acid that's 1/3 of the proteins involved in the amino acid sequence, and how proline and that unique amino acid hydroxyproline is present. And we have that very unique amino acid sequence, and we go down to the 3-chain coil helix, and then we see the final protein, which is tropocollagen, and we put tropocollagen into a matrix to form a collagen fibril. And there's that quarter overlap of tropocollagen with the tropocollagen next to it. We kind of ended there. We talked about how the collagen fibril, how it looks and how it was made up. And then I gave you these other pictures to kind of help you to see how that single chain molecular helix went together to form the 3-chain coiled helix, and how that structure allowed for elasticity. So the next thing I want to talk about is, how do we make collagen? We know that there's the fibroblast, where the cells in the pink-purple slide, we saw a nuclei. We know that there are cells that are embedded in the ground substance in between the fibers. And this is a good picture of a fibroblast. A fibroblast is differentiated from a mesenchymal cell. And this is the picture from the book, and then we'll talk about this as just kind of a duplicate information, just presented in a little bit different form. To produce a tropocollagen, the fibroblast, when given the signal that new tropocollagen is needed, either repairing a tendon or repairing the connective tissue matrix, the fibroblast actually produces a protein called procollagen. Procollagen differs from tropocollagen in that it has extra side chains on both ends of the protein. Those side chains allow procollagen to be excreted from the fibroblast into the extracellular matrix. So you can see here where procollagen is being transferred across the cell membrane. Those are called alpha chains. And then once the procollagen is in the extracellular matrix, there is an enzyme called collagenase that clips off those alpha chains to make tropocollagen. And the tropocollagen then are the proteins that are placed into the fibril matrix that we've already seen. So you need to remember fibroblasts are there, that it produces procollagen, then collagenase breaks off the alpha chains to produce tropocollagen, which then is inserted into the collagen matrix of the fiber. And this is the same picture, it's just showing you a little bit more detail as far as excretion of procollagen. And what I liked about this is it kind of shows how tropocollagen is put into the matrix of the collagen fiber out here in the extracellular space. So really that's what I wanted you to see here, is how tropocollagen then is put into this extracellular space, and that there's binding between tropocollagens to help maintain that fiber integrity. So that brings up to the topic of cross-linking. We've talked about the 3-chained helix that's used to make tropocollagen, and I used pipe cleaners and made some tropocollagen, right, in class. Here's my tropocollagen. I don't know if you can see it very well, but there are 3 pipe cleaners. There's a orange, a pink, and a green one. And we have that 3 chain coiled helix in there, and it helps it hold together. But what's interesting about this tropocollagen I made is I made it out of pipe cleaners. And the cross links, it doesn't need any cross links because the wire in the pipe cleaners have some rigidity, right? In reality, each of these helixes, single chain helixes, all I have here are a bunch of collagens, but each one of them is like a wet noodle. And if you try to put 3 wet noodles together, they don't bind together very well. We have to have something that's going to hold this matrix together. And we call that cross-linking. And there are 2 places that we see cross-linking. And the cross-linking that I'm talking about within the tropocollagen, within one tropocollagen inside, to hold those 3 helical structures together, is inside, intramolecular cross-links. And they're within the tropocollagen molecule. We also have, so we can bind the wet noodle together, the tropocollagen, but we have to put the tropocollagen, we make it end to end, right? Have 2 tropocollagens here together, and we make a line of those. Sorry, I'm not showing those very well. There, a line of those. And we have another tropocollagen in the next line above it that's offset by a quarter. And we have to bind those 2 together. That's what the last slide showed us. There has to be some cross-linking. That cross-linking is called intermolecular, between tropocollagen molecules. What I showed you in the previous slide right here is intermolecular cross-linking. Intra is within the tropocollagen molecule. And if I even go back to this slide, intramolecular cross-linking is right here within the 3-chain coiled helix, and intermolecular cross-linking is between the tropocollagen molecules, or proteins, to help form the fiber. Well, these cross-links are the same. There's actually a 2-stage cross-linking. There is an enzyme. It's called lysyl oxidase. Lysyl oxidase is involved in the 1st stage and also in the 2nd stage. Lysyl oxidase has 2 active sites. The first active site is that when you form a cross-link, which is between lysine and glycine, those cross-links are very heat soluble. At 60 degrees C, they will shrink and dissolve and form gelatin. And that all cross-links, whether intra or inter, are first formed with lysyl oxidase, and they're soluble cross-links. And the second step also uses lysyl oxidase, and there is an oxidative deamination. We're going to talk about that in a minute, with copper as a catalyst, and it forms a heat-stable cross-link, and it just takes that cross-link that's already formed and it changes its configuration, and it's no longer heat soluble. So the main points here is you need to know what that lysyl oxidase is involved in step 1 and step 2, that these cross-links can either be intramolecular or intermolecular. And the same thing happens when a new collagen's formed. The are within the helixes are soluble cross-links, and with time they turn to insoluble. And then when that tropocollagen's put into the fiber, the first cross-links to bind it in are soluble cross-links, and they're intermolecular. Well, cross-linking is very interesting because we've kind of talked about collagen as collagen only being one type. Now, I've only made, even though I make very bad little cross-links with my pipe cleaners, they all kind of look the same. But in reality, there are two main forms of collagen, type 1 and type 3 collagen. Type 3 collagen we actually find in very high prevalence in embryo development. It's smaller in diameter. It is less soluble because it has disulfide bonds, and as the animal increases with growth embryonically, we see the type 3 getting replaced by the type 1. And so it makes sense if you're going to build an animal embryonically, you want something tougher, that's smaller, that's going to provide that structural support, and then type 1 kicks in and starts to dominate. There are also type 4, 5, and 7. There are other types of collagen that differ slightly in their amino acid structure and how they go into fibers. Those are very minor roles. So as meat scientists, we mainly deal with type 1 and type 3. And we know that both of those types are going to have the inter and intramolecular crosslinks. And those crosslinks are going to first be lysine aldehyde crosslinks. And so that's between lysine and glycine to form a lysine aldehyde crosslink. That lysine aldehyde crosslink, remember, it was formed or catalyzed by the enzyme lysyl oxidase. Well, once that crosslink is formed, then lysyl oxidase has the ability on a different binding site to do an oxidative deamination, and copper is the helps to catalyze that, and lysyl oxidase is the enzyme that catalyzes that reaction. And so that there are two pathways, that aldeamine, where we get the lysine or hydroxylysine to form peptidyl aldehydes. That's a lysine aldehyde that we just talked about. Those are heat soluble. And then the second crosslink is the heat insoluble crosslink. And this is a ketoamine crosslink, and there is an amino rearrangement of that initial aldeamine crosslink to produce a ketoamine derivative. That ketoamine derivative is insoluble. So this is the first, step 1, this is step 2, both via lysyl oxidase. And step 2 occurs with more time. And if you look at, this is looking back at that initial picture, I wanted you to keep that in mind. And now look at this picture from Purslow. And Purslow is one of the papers that I gave you. And I gave you the paper because I want you to have a chance to look at it and try to really understand the crosslinks. Now, he talks about trivalent mature crosslinks and divalent immature crosslinks and glycation crosslinks. The point that I really want you to get to at this stage, because you'll talk about these other crosslinks later on if you're interested in more of the basic meat science, is that we can he introduces the concept of immature, mature, and then glycation. But each one of these is a tropocollagen. and he's showing the different types of cross-links and how those are he's only showing inter. Doesn't mean there aren't intra, but he's showing inter molecular cross-links. So you can see how it helps to hold that matrix together. Now he's using immature cross-links here are these ones like this, and then the mature, the trivalent ones are those thicker ones. And then he introduces the concept of what glycation is doing within the muscle. So there is the cross-link pattern. Always keep in mind, inter and intra do the same thing. The only difference is that both of them are going to do step 1, the aldehyde, and then step 2 is the ketoamine with the amira D rearrangement. And why would an animal want to go through a 2-step process? And we know that when animals are first born, their collagen is very elastic. So I think I've talked to this class about my puppies. I have little Shiners, and I have my big dog Shiner and Lacey, who are the parents. And the little Shiners are just starting to walk. And by day 2, we went in and had their tails bobbed off because they're Australian Shepherds, and that's what everybody wants. And it wasn't that big of a deal because their bones are mainly cartilage. And as they get older, they get stronger. So they're just a little over 2 weeks old, and right now they're learning to walk. And right now they walk like drunken sailors, and sometimes they have moments of of some coordination. But as their bones are developing, they're gaining strength, and their muscles are gaining strength too. So the mature cross-links in this case, and this is also from the Personal article that I gave you, and notice on the horizontal axis is time, so birth and increased age to physiological maturity. And physiological maturity isn't necessarily old, but just when they reach mature size. And in this slide, he had what he put is these dark lines, solid lines, are reducible aldamine and the oxoamine crosslinks. The dashed line, single dashed line, are the mature heat-soluble crosslinks. And then this dashed with dot line is the solubility. So that's the percent soluble crosslinks, the ones that are heat labile, will break down with heat. And notice that the percentage decreases with age, indicating that there's a higher percentage of the mature crosslinks, the heat-stable crosslinks. And that is true. You can look at those relationships and see how as you convert the heat labile to heat stable, or mature and immature, percent solubility goes down. And that's so that muscle, because we're just looking at paramecium here, and we're not looking throughout the whole body, we know that it's going to need to gain strength. And that's part of the aging process. That's why collagen has been studied so much, because it's an important part of the aging process. And we know that that crosslinks, as we look at young and old animals, and so this is cooking temperature, and that shear force value on the vertical axis, is that as we increase internal temperature, part of what happens is that meat gets tougher. And then, and so what this is really trying to show you, and there's some other things going on here, but in the young animals, we're going to have some swelling of the collagen and then shrinkage with heat. But what they're trying to show you here is that once you get to 60 degrees, the tenderness is much improved because there's solubilization of the collagen. In old animals, that occurs at 60 degrees, we start getting some solubilization, but we never get to the same amount of solubilization. The connective tissue never gets tender, as that from the young animals, because we can't get as much of the collagen to solubilize. So that's just to confirm the strength of single fibers and paramecium with Warner-Brasler shear force. So collagen fibers are going to change with age, and they're going to go through that two-step process, and that's going to happen at the intra and intermolecular level. Elastin is the second fiber that we can also find in connective tissue. Remember, we saw elastin in the pink slide as just little lines, right? Dark lines. Elastin is not as abundant as collagen. It's very rubbery, so it's present more so in ligaments and arterial walls and in the framework of organs. We also find it a lot of elastin in ligaments, and the cervical ligament that holds the head of the animal up in ruminants is a very large cable-like structure. It's yellow in color. It's called the back strap in layman's terms, and it runs from the head because it helps hold the head up, and so the head can move down and up as the animal chooses with muscle contraction. But we don't want to eat that because it's really, really chewy and it doesn't break down with heat. And so one of the things that you always do as a meat scientist is that for the chuck and the rib cuts that have cervical ligament runs along the top of the spinous processes, you always define that be removed. What's interesting about elastin is that glycine, just like in collagen, is the amino acid that is present at the highest amount. And for every 8 glycines in elastin, there's 1 desmosine and 1 isodesmosine. And the desmosine cross-links are incredibly heat stable, and desmosine is 90% nonpolar amino acid, so there's not much interaction with water. It's more difficult to solubilize. And some of those desmosine cross-links are going to also have sulfur cross-links, which are really, really tough. So desmosine and isodesmosine are unique to elastin. That's how we also measure elastin, is with those specific amino acids. Elastin is higher in the eye of round, which is also known